You're echoing. I'd like to welcome you all here today for our HIST webinar series, ChemHIST Talks. Our first speaker is Arwyn Smalley. Arwyn is a professor of chemistry at St. Martin's University in Lacey, Washington. Her PhD is in materials chemistry, but her interests are diverse. She regularly teaches general, analytical, and physical chemistry courses, but she also has taught special topics courses, including chemistry in the kitchen, the chemistry of food and cooking, atomic narratives, a class she co-taught on chemistry, ethics, and history of the Manhattan Project, and most recently, chemistry and art, focusing on the chemistry of art materials. Most years, she gives presentations at American Chemical Society meetings, usually on chemical education topics. This is her first ACS talk on a history topic, and we hope it won't be her last. So if everyone will mute their speakers, we will let Arwyn start.
All right. Thank you so much for tuning in today. I'm just going to share my screen here and minimize myself. Okay, so to give you a little bit of context here, this is a project that came out of a class I took as a student last year. So I took a Pacific Northwest history class, and one of the assignments that we had was to do a research project of any kind, any topic we were interested in. So I have lots and lots of things that I'm interested in. I could think of all kinds of different things, but I thought, you know what? A while ago, I remember going to the Burke Museum in Seattle, and there was this painting that I saw, and on it, they had a little detail that said that the Coast Salish people used these woolly dogs that they had raised for this purpose to make wool out of their fur. And so you can see the woman weaving here, and that the woolly dog is now extinct, but they also use mountain goat wool. So this stuck in my head from however many years ago that was. And as a knitter, I thought, I wonder if I can find anything out about that, because this would be a history project, right? And so I thought, well, I guess we'll just jump right in and see what happens. So like most research projects, you never know what is going to happen in terms of how much, how many resources you can find about it. But because I know there's a pretty wide variety of folks in the audience, before we go any further, let's talk about what I mean by Coast Salish. So the Coast Salish people are folks who have since time immemorial existed in the Pacific Northwest area. And this is the traditional area shown in the bright blue for the Seattle general vicinity, and then up into Canada and on to Vancouver Island. But to give you a little bit more context here, when we say Coast Salish, there's sort of this implication that this is all one group of people. It's really not. So there are a wide variety, as you can see, of a bunch of related language groups, and these are shown in colors. So this is the Coast Salish language group, which are still fairly diverse people. And then in the gray areas around them, there are still a bunch of folks who were living in these areas and still live in these areas too. So these are the traditional areas of the Coast Salish, and then other related or less well-related tribes in the vicinity. Okay, so coming back to the research project, when I first started looking into this research, one of the first things that I found was this book, Salish Weaving. So I found references all over the place to this book. So requested it from the library and started reading it. And Paula Gustafson wrote this book in 1980, and she did very comprehensive historical research looking at these textiles. She is a weaver and a fiber artist. And so she was looking at very closely at the styles, at the different changes over time, and then trying to look at the materials. And so she went through and talked about the fact that there is in the record this documentation that the Coast Salish used these woolly dogs, but that she couldn't find any reference to or any evidence of these materials when she was looking at them under the microscope and visually. So this was, it seemed like, uh-oh, did people actually use the woolly dogs? Was this just sort of a sideline and it was overestimated? So here's where I really started looking at the history research side of it. So for those who are new to history, like me, history research looks a little bit different from chemistry research, but in a lot of ways it's very similar. What we're looking for are the primary sources. So when I do chemistry research, I'm going to the journals where I'm looking for what are the articles for people who have actually done this work, right? And what did they do that worked? And what did they do that didn't work if they talk about it? And can I replicate it? And so in looking at the historical research, what I'm instead looking for are some of these early resources that are primary sources. So what were the people who were actually there saying about this? So we actually have to go back to the third Cook expedition to find the first English language reference to people going into this area. So Cook himself did not talk at all about the dogs, but John Ledyard, who was on the expedition as well, kept a journal, and he noted in his observations that the native people, their clothes were, some of the styles were made from the hair of their dogs, which are mostly white and of the domestic kind. So this is our earliest written record of the dogs showing up and people actually saying, yes, they're using them for their fibers. So fast forward a little bit to the Vancouver expedition, which went all along the West Coast from what is now San Francisco up to Alaska. And they spent quite a bit of time in the Salish Sea. And in Vancouver's journal, he also described the woolly dogs, and he was a little bit more comprehensive about it. So to highlight a couple of these areas here, he says that dogs resembled those of Pomerania. So this is a Pomeranian, in case you are not familiar. And he says they were a little bit larger, but think about that fluffy shape, right, with the curly tail. He said they were shorn close to the skin as sheep, and that the fleeces were compact. You could lift them up by the corner, and they were made of a mixture of coarse fibers and very fine wool, which could be spun into yarn. So here we have two early, late 1700s examples of primary source journals where people are talking about their eyewitness accounts of seeing the native people using dogs in this way. So I just went along with the assumption that this is probably, these are credible sources, right? And there are actually quite a few other resources that talk about this. But the third one that I'm going to do is coming back to this painting, which was by Paul Kane in approximately 1856. He did a lot of painting. He was an artist who was traveling through Canada and spent quite a bit of time living near the native people in a wide range of different locations across Canada. And one of these was in the Fraser Valley in British Columbia. And so he did this painting that I had seen in the Burke Museum, although, I mean, that one's a copy. And in his journal, he also talked about the dogs. So he specifically talked about the same thing that Vancouver did, that they have these dogs, they are white or brownish black, and people are using their hair for spinning. And so early on, the dog that you see in the foreground, which is white and fluffy, was identified by a lot of folks as, oh, this is probably the dog he's talking about. But he actually did drawings in his journals that are probably much more accurate for what these dogs looked like. So we're getting this sketch of, again, the curly tail and the fluffiness of these specific woolly dogs. And we also have in the historic literature lots of references of intentionality, that you have people keeping these dogs away from all the rest of the dogs. They would have these flocks of dogs. They might keep them on a small island right off the coast so that they couldn't intermix with the other dogs that they would have in the community, because they had hunting dogs and, there's a variety of dogs in the village. So you have lots of historic research that points to, yes, there are these intentional dogs that are bred for the purpose of producing hair. Okay, so now this is all very history so far. And now coming back to Paula Gustafson, who wrote in her book that she didn't think that people had used the dogs, but let's give her some benefit of the doubt, right? She's looking through a microscope, she's using optical methods, she's looking at fibers that are 150 or 200 years old. Some of these are very degraded. It's also very hard to tell one fiber from another just by looking at the outside of it, especially when it's degraded. There weren't a lot of tools that were scientific at the time to do any analysis on fibers. And it turns out you can't really do any genetic analysis on these unless you have the cuticle of a hair, which doesn't come along when you're just shearing the hair. So, I mean, once you think about the hair itself, it's just protein, and a lot of different kinds of protein are pretty similar. So, turns out I was not the only person who was curious about this. So I looked further into the scientific literature because now, right, we're going to get into the chemistry side of this history project. And so I found a really clever analysis looking at some burial goods that included a blanket that was believed to have the dog hair in it. So this is a find that is up at the Simon Fraser Museum of Archaeology that's in British Columbia. And this work was done by Dr. Schulting in 1994, who was there at the archaeology department. This Yale blanket was found as a grave good, so it was wrapped around the body of an infant that was then put inside a container that was made of bronze. And so because of the copper in that container, you had exceptional preservation of this blanket. And so at the same time, you also have all kinds of staining. It's probably green. And so you can't really do a lot of visual analysis on it. But he had good reason to think that this might be an example where he could find some dog fibers. So rather than, again, this is 1994, he doesn't have any cuticles, he's just looking at the proteins, but he used some very clever archaeological tools to do some deductive reasoning here. So what he said was, rather than just looking at the fibers, let's look at stable isotope analysis of these. So this is a very common tool in archaeology, and what it is looking at are these stable isotopes, usually carbon, oxygen, and nitrogen. And it's looking at the isotope ratios. So, everybody knows about like radiocarbon dating, right? Those are using radioactive isotopes, but these ones are just the regular stable isotopes. It turns out that the isotope ratios vary a lot based on plant metabolism. So you have two main routes of plant metabolism, and they create different carbon ratios. And then depending on, and these ratios are listed as delta values, and then depending on what an animal or a person is eating, then the ratios are going to get modified in that organism. And so depending on diet, so if you have an animal that just eats plants like a rabbit or, a mouse or something, they are going to have a particular delta carbon ratio. And then if you have another animal that mostly eats those animals that are eating those mostly terrestrial food sources, they also will have a very similar delta carbon ratio. On the other hand, if you're eating a lot of either freshwater or marine fish, you'll get different carbon isotope ratios. So Dr. Schulting said, well, hey, look, if I have a domestic dog, it's probably eating the same thing, more or less, that its people are eating. And the Coast Salish people had a very high marine-based diet. And so they would have a different delta carbon ratio compared to an animal like a mountain goat that would be eating primarily terrestrial food sources. And there's about this 7% difference in the delta carbon between these two. So it's distinctive enough that you should be able to distinguish between these. So, and this is, again, very commonly used in archaeology to look at trends in diet in different populations over time. So he looked at the stable isotope analysis of the blanket in several different locations. And so these data are shown on table 1 here. And so samples 1 through 8, if you take a look at these for a moment, the numbers on the far right, these are the delta carbon ratios. And so you can see that all of them except for this sample number 4 are all around the -15 range, -15 to -16. And then this sample number 4 is a -20-ish. And then the comparison samples are down below. Those are 9 through the following. There's a bunch of numbers there. Mountain goat, these are contemporaneous, or excuse me, those are contemporary animals. So this is not a historic mountain goat, but you figure probably mountain goats are about the same over time. So that one's coming in at a -21. The coyote, the hare and the bone are both from the same animal. And you can see a little bit of variation there. This human infant metatarsal, that was from the buried infant that presumably would have been in the same community. And you can see that that number matches up really well with all of the blanket samples except for that number 4. And then the deer hair was included, the deer and the coyote and the mountain goat. These were the 3 fibers that Paula Gustafson included as possible alternatives. So we can see from these data that there is a very good match between the delta carbon ratio for the human metatarsal and all of those samples in the blanket except for that sample number 4. So they redid that sample number 4 just to check on it, and it was confirmed to have the same number, which they concluded to mean that it was probably mountain goat or something else, that they ran out of the dog fiber, and so they were using a little bit of something else. So this is our first definite evidence, if not, it's not definitive, it doesn't prove that it was dog hair, but at least in terms of the simplest conclusion that we can come to, it makes the most sense that these samples do actually come from a domestic dog. But of course, we want to continue to have a little bit more proof here. So time moves on and technology moves on. And so the next set of analyses was the Salaza group in 2011, and they looked at protein mass spectrometry. So they were looking at the fibers, and the proteins have been characterized fairly in good detail, at least for a domestic dog. And so this protein mass spectrometry is effective at distinguishing between these protein sources between animals, or at least animal groupings. So they analyzed 10 different Coast Salish blankets of different styles and time periods. And then they also were lucky enough to rediscover that in the Smithsonian collection, there was this pelt, which you can see on the bottom right of the slide, from the one known, or at least suspected, woolly dog sample that exists anywhere, which is in the Smithsonian collection. We'll talk more about that later. So the protein mass spectrometry, this is a very busy slide, but over here on the left-hand side, you can see the dog protein. These are coded. And then the second column over, this is the Salish dog. And that's that pelt. So you can see it matches up in a lot of ways, but there isn't a perfect overlap. The dog protein on the left, this would be for modern dogs. And so you do end up with some changes over time. So the flurry of X's over here on the right-hand side, these are from the 7 blankets out of the 10 that they analyzed that had dog fibers in them. And these 2, the 1892 and the 2124, had areas of all dog fiber, and everything else there was a mix. So if we look at all of those blankets over time, this is a little graph showing the top 3. You can see on the left-hand axis, this is our time for the collection time for those blankets. So those last 3 are later than 1900. And according to the primary source literature, all of the dogs had gone extinct by about the 1860s at some point, maybe a little bit later than that, but certainly before 1900. So those 3 blankets not having any dog fur is totally consistent with what the primary source records indicate. So those 2 samples that had areas with just dog fur, these are these 2 in the red box now, and that was pre-1850. So again, that timing is very indicative for this is toward the end of the time period when people were keeping these dogs, but still within it, right? So we finally do have some pretty definitive proof, right? We do have dog fur that is definitely happening in these blankets. Now, the next question is, do we have any evidence that these are a distinct breed of dog, or some people in the historical record were suggesting, well, maybe these were imported from somewhere. Maybe there were some, I mean, there's some pretty wild hypotheses here. Maybe there was a shipwreck from Japan and people brought over dogs, I'm not kidding, and brought over dogs from there. And maybe this is where that population come from. So let's come back to this woolly dog pelt. So back in the early 2000s, actually, there was a beautiful ethnographic study where they located this pelt and had asked for some people to do genetic analysis of it. This was very early in that time period when you had it available as a tool. And in the article, very tantalizingly, they were like, we've asked people to do this analysis, but we haven't gotten the results yet. And I was like, ugh. But then December 2023, there was an article published, and so this is, 3 months before I'm doing this project. There's an article published in Science that did a complete genetic mapping of this exact pelt. And much of that information, I'm not a biologist, so much of it was way above my head. But let's kind of jump into the short version of that story, which is one of the things that they analyzed was the mitochondrial DNA, and this allows them to look at genealogy and the consistency of the genealogy. So this, again, is a very busy slide with a lot going on. So what we look at over here on the left in the turquoise is this is a genetic map for where our common ancestors are. So they're grouping the animals by clade, which is a group of animals that have a common ancestor. So you can see all the ones in turquoise have a common ancestor that goes back somewhere between about 2,000 and 5,000 years. And they all are in one group together with a bunch of other animals that are in the northern part of the North America. And then also there's one dot over there in sort of northern, Eurasia there. And then we've got another set of more closely related animals that are in the green that are further down into the North America. And these ones all have a specific protein, a specific DNA, the A2B trait. And this one developed after the dogs migrated. And so these are not shared with the warm colored dots here, the red and the yellow. Those ones are European breeds, and those ones have diverged in their DNA. So all of the A2B dogs are in one group together, and then all of the other dogs are related to each other. So what this graph tells us definitively is that mutton was an example of a dog breed that had been intentionally bred in the Pacific Northwest or in the Alaska area as well, depending on, migration patterns, that this was an animal that was kept for this specific purpose. It was intentionally bred over a long period of time and was a distinct breed. And so this matches up with all of the historical data that you not only saw that I included, but also all of the oral tradition for the Pacific Northwest natives who have in their historic, in their traditional knowledge that this has been true, right? And so Dr. Lynn and her group actually collaborated with a number of First Peoples and Native Americans in the Pacific Northwest to make sure to take those stories, the traditional stories and histories, the oral histories, into consideration when looking at this research. So this was a collaborative work. It's a pretty interesting article. Highly recommend that you check it out. So we have not only the proof, right, that we were looking for, that this dog was used for fiber, but also that it was a distinct breed. And now, of course, we have one more important question, which is, but what was the dog like? And luckily, they actually did some like artist impression, right, of what would this woolly dog have been like. And so we have a medium-sized white fluffy dog that looks very huggable. And we have it on this map, or this little chart, with a bunch of other contemporaneous breeds that are still in existence. So if you have a huge craving to own something like a woolly dog, probably the American Eskimo dog is one of the closest ones, a little bit smaller. Maybe a Finnish Spitz would be a good choice as well. So I have included most of the resources that I used. There maybe were a few that I didn't end up citing in the article, but which I used for part of the background research. And then I really appreciate my teacher and former colleague, Erin Goings, who taught this amazing class, and my faculty development committee for helping me get to the ACS. And of course, all of you, thank you. And if you have any questions, I'm happy to take them.
Thank you very much, Arwyn. That was a great talk. Does anybody have any questions? If you do, just unmute your mic and ask away. I guess another
Hi,
question.
my name is Nelly. Nice to see you. Great presentation. I really enjoyed that. I wonder from a technical viewpoint, the wool seems, the fibers don't seem to be very long. It takes probably special kind of technique to make to spin it, to make fibers. I can't even imagine how from this type of wool they created whatever they created from it. Do you know some details about the technique they used?
So my understanding from talking to people who have huskies and similar kinds of dogs is that you have actually some fibers that are fairly long, and I mean, these fibers are not going to be like your sort of standard single coat dog where the fibers are very slippery, but they're going to have a double coat where one of them is fairly rough and then the other one is longer and much silkier and thinner. And so those ones are, once you separate them out, they're going to be able to be spun together using a standard technique. And if you look on the internet, like there's people all over the place who are, I made a sweater out of my Malamute fur. And so you will find people who are doing that even now.
Very interesting. Thank you.
No problem. Thanks for asking.
So Arwyn, is Ms. Gustafson still alive?
I don't know. I haven't actually checked.
Because I wondered if you were going to make an attempt to let her know that the research has gone past what she'd
That's a
written.
really good question. I hadn't even thought to look that up. I hope that whoever did that the Lynn group maybe had a conversation with her about it. But there was definitely in the probably 20 or 30 years after the 1980 book, there were quite a few articles that sort of addressed her directly or her research directly, like especially the Schulting referred to her book quite often. That also is a fabulous paper, by the way.
Okay. Any other questions?
I've got a question. This is Mary Jo, also at St. Martin's, actually right down the hallway from Arwyn.
Hi, Mary Jo.
I was just a little late. I had some issues with my computer, so you might have said at the very beginning. But did these become extinct, or they just became hybrids with something else, or do we even know?
Yeah, so basically probably both. And the why for this has to probably do with a couple of different things. And unfortunately, I suspect that one of them is a lot of cultural suppression, that in the 1800s, when the treaties were first being organized with the Pacific Northwest tribes, that tribes were not allowed to do their traditional practices, including things like keep dogs. And so this would have been something that it was a women's job to have in fact, it was a very high prestige woman's role to have a big group of dogs. And the women in the historical record since then have talked about, yes, my grandmother used to keep these dogs, and then we were not allowed to keep them anymore. And so it was largely, the cultural suppression that happened. And then dogs, if you're not taking care of them, they're just going to interbreed with everything. So there are probably some dogs that still have some of this genetic material, and that could be found, but not as any distinct breed.
Okay, thank you very much, Arwyn. And we will now head on to our next speaker. So our next speaker is Nick Tsarevsky. He got his master's degree in theoretical chemistry and chemical physics in 1999 from the University of Sofia in Bulgaria, and his PhD in chemistry in 2005 from Carnegie Mellon. He joined the Department of Chemistry at Southern Methodist University, SMU, yes, Dan, in 2010 and was promoted to associate professor in 2016. Since 2022, he's also been an affiliate faculty in graduate liberal studies in the Annette Caldwell Simmons School of Education and Human Development at SMU. His research interests include controlled radical polymerization, development of efficient polymer functionalization reactions, hypervalent iodine chemistry, chemical education, and the history of chemistry. He is the author of many books and journal articles. He's also the author or co-author of 3 science education scripts, 2 for TV and 1 for radio. He served in HIST as a secretary in 2005. Oh, wait a minute, that's wrong. He was secretary in 2005 and chair of the Polymer Group of Pittsburgh local section of the ACS. He also served as chair-elect in 2008 and chair in 2009. He served as program chair of the HIST division from 2018 to 2024. He's currently chair-elect, and he was awarded the Wilford Doherty Award of the Dallas-Fort Worth section of the ACS in 2020 and elected an ACS fellow in 2024. So he's going to be talking about Torbern Bergman. So I turn it over to Nick. Thank you.
Thank you, Vera. Thank you very much. I hope everything is visible and I'm audible. I will start by thanking Gary Patterson, who organized the symposium at the last ACS meeting on the history of chemistry in the 18th century. So this was a talk given as part of that symposium.
Nick, can you go to slide mode?
Yes. Is it okay? Can you?
There you go.
Okay. Yes, some delay apparently. Yes. So in addition, I would like to thank Anna de Bettencourt Diaz and also Vera for making all these talks possible. So this is really delightful to be able to give this talk. I'm very honored and happy to do that. So as Vera said, I will talk about a chemist famous at the time, Torbern Bergman, a Swedish chemist, who is perhaps not necessarily in need of resurrection. He's still known to some degree, but he needs to be known better. And so I'll give a talk. It will be really in the form of, I would say, snapshots of most important things he did, because he did quite a lot. Let's start from the very beginning. He was born in 1735 in March. The date is not exactly certain. His father was a tax collector or revenue officer, Barthold Bergman. He started his schooling at home, in fact, and so he was educated at home and was reading a lot of books on mathematics and philosophy. Botany was particularly important topic to him. And so in 1746, so after some homeschooling, he attends the school in Skara and really started collecting and studying the plants that were growing near that area. And by the way, there is a little map right here on the side. So just to give you some of these, so this is Skara. It's shown on the map, just to have an idea where he was. Uppsala is where he went next. That's in 1752. He started studying there. His father pushed him to study divinity or law, and Bergman absolutely hated that. He really was interested in mathematics, physics, all the natural sciences, botany, entomology, and there were some friends there who attended the University of Uppsala, friend or cousin, perhaps, some relative, who in fact at one point confiscated Bergman's books on physics and mathematics and all the natural sciences just to push him to study law or divinity. Bergman got apparently very sick after that, I suppose some kind of very severe depression, and so he had to return home for a while. And apparently while he was there, he managed to convince his parents that, you know what, I am interested really in botany and mathematics and in the physical sciences, not in theology. So he was allowed to go back to Uppsala and actually to study what he really wanted. So in the next decade or so, in 1750s, 60s, he works in all kinds of fields of natural philosophy, mostly biology, but also physics. He actually showed that there was a little organism that Linnaeus, the great Linnaeus, who was a patriarch of botany in Sweden at the time, had thought to be a female coccus. It's a type of insect. But Bergman, in fact, shown that it was ovum of a leech. And so he showed his work to Linnaeus, and Linnaeus was a little bit hesitant to admit his mistake at the beginning, but was convinced at the end. And in fact, he wrote on his on Bergman's paper, "Vidi et obstupui." I saw and I was stupefied, amazed, right, by this work. So very high praise from very important person. Here is Linnaeus, of course. He's famous for his taxonomy of plants and animals, the nomenclature that we still use, genus and species, for all living organisms. He did other work as well. I mean, he worked in Sweden. There is a famous scientist in Sweden, Celsius, who developed temperature scales. So Linnaeus actually worked on that as well. But he's really most famous for Systema Naturae, the system of nature, that book with nomenclature, which in fact influenced also the chemical nomenclature, which was developed a little bit later. So he was a supporter at the beginning. They had some frictions with Bergman later, but at the beginning, he was an ardent supporter of Bergman, which really helped. So in the late 1750s, so in 1758, Bergman publishes a paper on universal attractions with very heavy on mathematics. He really knew mathematics, and he wrote a lot of equations, solved the various types of attractions, universal, as he said, so that includes gravity, but other things as well. I'll talk a little bit about it later. He sent a couple of letters to Wilson, which were published in the Philosophical Transactions on electricity. So he studied that as well. He studied the aurora borealis, for instance, all kinds of physical phenomena. Did a little bit of work on astronomy as well. In 1761, he was appointed magister or master docent in physics. Continued to work on electricity, and particularly electrical properties of the Iceland spar, and later on the tourmaline, which I'll mention as well. By 1767, there was an open position as chemistry at the University of Uppsala, which was left vacant by Johann Gottschalk Valerius, and so Bergman applied for that. I have to say here that many of the things that I will be mentioning, some original papers, I'm very much obliged to this bibliography that was published quite a few years ago. So his complete work, it made it really easy to find original sources. So I have to acknowledge a bibliographer who collected all his work. Okay. So Valerius was the professor up to 1767 of chemistry at Uppsala. That's him here shown. And he was Swedish brother of Niels Valerius, who was a famous physicist. He studied at Uppsala, got his master's degree in mathematics and physics, and then eventually a medical degree at Lund. And so between the years 1750 and 67, he was a professor of chemistry in Uppsala. Mostly interested in mineralogy, and this is a book which was clearly very widely respected on mineralogy, because you see various translations. That one in the middle is in Russian. So even in Russian, in 1763, it was translated. So it was quite a popular book. Also a book on agriculture from a chemical viewpoint. But in 1767, due to poor health, and by the way, he managed to outlive Bergman, but he retired in 1767, and the seat was left vacant. So by that time, Bergman applies for that position. He had written only one single paper on chemistry, and it was a work on manufacture of alum. Valerius wanted, in fact, one of his relatives or one of his students, not exactly clear, but somebody he was connected to take the position, and really viciously attacked Bergman, criticized that single chemical work by Bergman. It wasn't good enough, so they became, I don't want to say bitter enemies, but there was quite a friction there. Luckily, Bergman found another huge supporter. He's shown here with his brothers. So this is the crown prince at that time of Sweden, the future Gustav III, the king of Sweden. He was in fact the chancellor of Uppsala University, so he had quite a lot of power and he had the say there who gets appointed at various positions. He decided to consult two mineralogists or mining engineers, Anton von Swab and Daniel Tillis, about Bergman, because Bergman was applying. They both had read his work on alum and some of his physical work as well, on electricity, aurora borealis, all these phenomena, and they very hard enthusiastically recommended Bergman, at which point, of course, the future Gustav III said, okay, well, he will be the future professor of chemistry. So he was a huge supporter of Bergman for a long time. In fact, in 1776, at a time when Bergman was invited to go to Prussia by the King of Prussia, Frederick the Great, to be a professor there of chemistry, Bergman refused to go, and as a reward, Gustav, at that time already king, knighted him. So he became the Knight of the Order of Vasa. So he was a huge supporter of the arts, theater, opera, the sciences. He was assassinated in 1792, Gustav III, that is. And those of you who like Verdi's music, next time you hear Un ballo in maschera, the opera, that's inspired by that murder of King Gustav III. So Bergman now has two important supporters, Linnaeus, the scientist, and of course the future king, who is the chancellor of the university. So he gets the appointment. This is that very first chemical work, by the way, that he published. So this is on the manufacture of alum. When I looked at that original document, it was very interesting the way he cites his sources. He mentions a lot of scientists. He actually made no discoveries here. It's a sort of review paper of what was known at the time about the manufacture of alum. So no original findings, particularly some importance. He cites a lot of previous work, of course, as he should have. It's interesting to note that he was clearly, I mean, to me, I'm not trying to get into his psychology, but the way he quotes Valerius, the previous professor of chemistry, is very flowery, and he cites a lot of famous great scientists there, the likes of Marggraf, Pott, Leibniz, Brandt, Boerhaave, Pierre Maquet, Geoffroy, Caspar Neumann. All these scientists are cited just by name. However, if you look here how Valerius is cited, the celebrissimi Professor Valeri, right, the most celebrated Professor Valerius, then there is the outstanding celebrated Professor Valerius. Nobody else is celebrated, but Valerius is the only one who these titles are attached to him. I don't know if he was trying to make friends with him or not. It certainly appears this way. Anyway, that's the work that really started his chemical career. Now, he became very interested in chemical analysis very soon. This is an early portrait of Bergman by Ulrika Pasch. There is some argument it's probably not a very good likeness, but a famous portrait of Bergman. So let's see what he did in the field of analytical chemistry. He really revolutionized it. There were quite a few color reactions at that time that were known for the analysis of mineral waters, salts, acids. Many were developed by Robert Boyle in the previous century, improved by the likes of Friedrich Hoffmann in Germany, but Bergman really did quite a lot. So analysis of mineral and sea waters. So he developed a very important, a very systematic methodology how to analyze these waters. First of all, he would expel dissolved gases, things like carbon dioxide, hydrogen sulfide, by heating, and then those gases would be collected. Then the water, mineral or seawater, would be evaporated, and then the residue would be extracted with a series of solvents, alcohol, water, acetic acid, hydrochloric acid. And then each of these portions, extracts, would be treated with various reactants. About 25 of them are described. Syrup of violets for pH, ammonia, sulfides to precipitate various metal ions, sodium chloride, HCl, to precipitate silver if it's present, infusion of galls to determine iron, lead acetate, silver nitrate, quite a few others. Oxalic acid for the detection of calcium was something that he introduced, as well as barium chloride. So this is another reactant for determination of sulfates. Many mineral waters contain sulfates, of course, and so this barium chloride, or salated terra ponderosa, heavy, salted heavy earth, was introduced by Bergman, it seems like. Analysis of minerals as well. He did quite a lot in that area. So his strategy was he would dissolve a mineral in strong acid and then proceed as he did with the mineral or seawaters. Not only quantitative, but also qualitative analysis were developed by Bergman. I have to say that if you look at his data, actually, it's never really accurate, particularly. But what he did, he introduced methodologies for analysis, and that's his main contribution. Very, very new and very systematic. The numbers should always be taken with a grain of salt. So here is just an example. He studied various compounds and their composition. So he stated, I mean, there is no need to always isolate a pure metal, for instance, to determine how much metal salt is present in some mineral water. All that needs to be done is precipitate it as some kind of insoluble material which has known composition, filter it, dry it, weigh it carefully, and then determine how much metal was there. And so he has this table, this is a translation, English translation from 1784, of various types of insoluble materials. So for instance, 100 parts of lead is precipitated by that amount of hydroxides and that amount of sulfides or sulfates, and so on and so forth. So for all these metals, he basically says how much metal, this is the number, 100 parts of these metals, in how much of the precipitated compound. So you can determine by gravimetry the amount of metal that was present there. More on analysis was done by him. So he's famous for really, I shouldn't say pioneering, but he was one of the great proponents of the blowpipe as an analytical instrument, very important analytical instrument at the time. It was really probably developed first in Germany by Johann Kunkel, famous for his work on glass making. But in that book on glass making, Kunkel basically mentions the blowpipe. So it's a little pipe that you blow at the flame to make it stronger and hotter. And he describes actually how this blowpipe can be used. So here is from his book, quotation. Many possibilities are concealed in this art with the blowpipe. In a chemist's workshop, for example, it might prove to be very useful. One use would be to melt a metal lime, so this is a metal oxide, in order to see what metal it contains. It is necessary only to place the lime to be analyzed into a hollow in a small piece of charcoal and then blow the flame onto it. So blowpipe was used for analysis as well. But Bergman really did a tremendous amount of work, perfected it. He had new designs actually of the blowpipe. He published a whole dissertation, was defended under him on the blowpipe. And this is just the table of contents of this dissertation. So as you can see, some of the chapters, by the way, are misnumbered, but that's in the original book, that's how they're numbered. But he talks about the instrument, how to make it, how to blow through it, how to use various types of fluxes to make things easier to melt, what should be observed. And then he analyzes fossils, salts, acids, bases, various types of earths, or as we would call them today, metal oxides, flammable substances. And so all these things were really carefully described. So really pioneering work on the use of the blowpipe, very important analytical instrument. At the time, there weren't that many analytical instruments, we should remember. So here is a picture from that book, and this is the flame. He identified that there are various parts of the flame. Some are, as we would call them today, more reducing, some are more oxidizing. He was aware of those properties of the flame, not only just the different temperatures. And he knew how to manipulate with the blowpipe to make hotter, cooler flame, more oxidizing, less oxidizing. And in fact, here is one application. This is a work, by the way, published by one famous, more famous probably now chemist than Bergman, Carl Scheele, in which he described the discovery of tungsten. And so this is a work by Scheele, published in 1781. And he talks about tungsten, by which he means not the metal, but he means actually the mineral, calcium tungstate. And he described how that mineral can be analyzed with microcosmic salt, he says, Scheele does. This is sodium phosphate type of salt. It's a nice flux. Tungstates give sea green glass. In the extreme point of the flame, the globule loses its color. The same effect is caused by saltpeter. So this is the oxidizing flame, which of course, when you add an oxidant, you get the same effect when you add potassium nitrate. But the color then returns if the blue part, the reducing part of the flame, is driven on the globule. And he describes various experiments here. He isolates tungsten trioxide, for instance, turns reddish yellow with the blowpipe and then brown. But then Bergman steps in, and so right after this paper by Scheele, Bergman essentially kind of to endorse, I suppose, the paper by Scheele, he not only says, yes, this is a very good paper, but he also does additional experiments. He is now, of course, the expert of the blowpipe. So with microcosmic salt, tungsten gives a celestial blue glass globule with no redness by refraction, which is produced, for instance, by cobalt. So he now distinguishes how to say, well, this is tungsten or this is cobalt. With more tungsten mineral, the globule becomes brownish, but still transparent. Molybdenum compounds, which were just barely known at the time, gives microcosmic salt a green color. And borax saturated with it appears gray in reflected rays, but dark violet by the refracted. So now he has also distinguishing features of molybdenum and also tungsten. So this is Bergman's contribution to analytical chemistry using the blowpipe. He's perhaps now most famous, I should say, for his work on chemical affinity. And this is his most famous well-known portrait by Lawrence Pash. In this portrait, he's holding right here a roll of his table of chemical affinities. So what are these chemical affinities? Chemical affinities have been studied prior to Bergman, but I would say that his interest in chemical affinities probably started with that paper that I mentioned, the universal attractions in nature. And so this is a very heavily mathematical paper, heavy on mathematics, a paper talking about various types of attractions in nature, such as gravity and so on and so forth. But he also mentions, well, fluids, for instance, droplets, when you get them close together, they merge into one. There is some kind of attraction. So probably these ideas started at that time. This is 1758. That's quite an early paper by Bergman. He doesn't talk much about chemistry here, but the universal attractions. Now, as far as chemical affinities, the most famous first work on chemical affinities was that published by Étienne-François Geoffroy in France. And he presented this table of chemical affinities. It's quite limited. There are very few. These are chemical substances. For instance, right here, this is that square or is mercury. And so what this is telling you is that the next thing closest to mercury, this is gold. That means that gold has very high affinity for mercury. Silver, the moon, next one, a little bit less of an affinity. And then the other metals, things like tin, copper, and so on, lower and lower affinity. At the very end, this crown here represents antimony, which has the lowest affinity for mercury. So this is how you read that table. So anything that the closer something is to the chemical on top, the higher the affinity is. But as you see, very, very limited. Now, what Bergman did was he enriched it immensely, and he published this actually table of chemical affinities. Not only is it much bigger, much richer, but he also distinguished two ways to perform chemical reactions. And he says, well, affinities or attractions between chemicals depend actually on how you do those reactions. So you can do them in the humid way or in solution, or in the dry way via Sitka here, as it says, right? So you can basically have two powders and heat them to high temperature. The affinities are different than those in solution, sometimes at least. So he distinguished single elective attractions. So we have A reacting with B or C with D. So which one, or does compound A prefer B, C, or D? Then he also talks about double attractions. So these are exchange reactions, as we would call them. A, B reacting with C, D, basically swapping their building blocks if the affinities are high enough. Discussed even a little bit about the impact of heat on elective attractions, effect of solubility, and all these things were discussed in this. So this is one thing that's very unique about this paper. He introduces these types of things. These are probably the first chemical equations, as we would call them, reaction schemes. These all represent chemical reactions. So for instance, The way he represents the reaction of calcium chloride and potassium sulfate to give you gypsum precipitate and potassium chloride in solution is by using their symbol. So this is calcium oxide, marinic acid, hydrochloric acid, in other words, potassium carbonate and sulfuric acid. When you mix potassium carbonate and sulfuric acid, you form potassium sulfate. This is one of your reactants here. And so this is the symbol of potassium sulfate. It's made by parts of sulfuric acid and potassium carbonate. And so here is his chemical equation or reaction scheme. So you have potassium sulfate when mixed with calcium chloride. They exchange their ions because calcium has bigger affinity for sulfate than chloride. So calcium sulfate precipitates, so it's shown here on the bottom. And in solution, potassium chloride remains. We call these potassium and chloride ions nowadays spectator ions when we teach general chemistry. So a very interesting contribution in representing chemical reactions. There was a medal, by the way, I collect some of these. So this is Bergman medal, which was issued by the Swedish Academy of Sciences in 1957. On the back of that, so there is a portrait of Bergman, but on the back, it's one of these chemical equations. So it's a very interesting contribution that he made. These inspired a lot of studies. I will not go too much into this because of time, but the Dictionary of Chemistry by William Nicholson uses similar types of representations. However, he tried to assign numbers to quantify affinities, not very successfully. These did not really work. These numbers were based on simply the amounts of compound A that react with compound B. And so these are not what determines activity. This is what we would call early stoichiometry. It has little to do with affinity, but certainly inspired by Bergman's work. He also did work on crystals and crystallography. He's probably less famous for that, but his early studies I mentioned, he studied the tourmaline, its electrical properties. He found out that it generates, produces, and holds electric charges when heated. We call this pyroelectricity, or when it's deformed or stressed. So some pressure is applied to it, which we call piezoelectricity. So that's a paper from 1766. This is the equipment that he uses depicted there. This is how his interest probably started, but he later contributed even more to that. Something that we would call today, when we're discussing crystal lattice structures, this is probably some of the work, the original work was done by Bergman. Now, sure, Robert Hooke in his Micrographia talks about how crystals are built, and he shows that close packing of spheres can produce various types of geometric shapes. So this is an early example of crystal lattices. Huygens, in his work on light, he has a very interesting depiction of a crystal, right? The structure of a crystal, right? Composed of molecules and atoms. But this is Bergman's work, 1773. He talks specifically about the primitive form. So this is a calcium carbonate, the spar mineral, and he talks about it. So primitive form, right? So this is the crystal lattice. It's rhomboid. Measured the angles, 101 degrees, 78 degrees. And then he also says that, well, by moving this simplest shape here up and down along this axis, you can actually generate the whole crystal. So this is the elementary a nucleus, as he calls it here, which actually can be moved around in space to generate the entire crystal. Now, in the last minute or two, I just want to say that Bergman was an ardent educator of chemistry as well, and this should not be neglected. So one of the things that he did first, there was a very, very popular lecturer of chemistry at Uppsala by the name of Schäfer, Henrik Theophilus Schäfer. His lectures were not really published. Various students had their lecture notes. So some notes from by attending his lectures. And what Bergman did is collect all these and heavily edit them with a lot of added comments. Right. And he published that. It was a very important textbook that was used and highly respected. It contains chapters on dyes and minerals and things of this nature. Lucky for us, this was translated actually in English, so it's available, the chemical lectures of Schäfer. So this is Bergman, the educator. He supervised quite a few dissertations, over 20, on things like fulminating gold, mineral waters, analysis, antimony, nickel, arsenic, various metals, asbestos, also the history of chemistry. So I just want to mention that two of the candidates that defended under him got their degrees. was Johann Afzelius, who was the future teacher of Berzelius, great chemist. So in a sense, Berzelius is Bergman's scientific grandson, as well as his brother Per Afzelius, who did a lot of work on chemistry and the history of chemistry as well. Bergman popularized chemistry. He published a very interesting essay. It's called Essay on the Usefulness of Chemistry. Right? So it was quite a popular. It's a very nice, just from the table of contents, he talks about salts and flammable substances, and also there is a little bit of agricultural chemistry in that. Very, very important book to excite the public and educate them about chemistry using simple language. It's very readable, I have to say. Afzelius, I just had to say, this is the history of chemistry division. So this is his work, the chemical progress in the 7th to 17th centuries, right? So this is Afzelius Arfvedson, who published a book on the history of chemistry. This is the very end of my talk. So he was really very highly respected. This is another medal of Bergman. He was member of the Royal Swedish Academy of Sciences and then the Academy in Halle. Also a fellow of the Royal Society in London, American Philosophical Society. By the way, this was published when he was elected a member of the Philosophical Society. It was published in the newspaper. It was seen as an important thing. This is the Pennsylvania Journal, or weekly advertiser from 1773. And so it mentions here that Dr. Bergman, professor of mathematics in Stockholm, became a member of the American Philosophical Society. Societies in Göttingen, Paris, that was very important because there were not that many at the Academy of Sciences in Paris. He was only one of 8 foreign members, and various others, right? Torino, Dijon, various scientific societies. So a lot of the things that I mentioned about Bergman was taken from some of those articles that were published after he died in 1784. So a lot of biographical information published by the French Academy of Sciences, and also the Journal de Paris. Actually, almost the entire issue, it's several pages only, but almost the entire issue is dedicated to Bergman's passing and his contributions to science. So I will stop here. I hope I was able to at least convince you that Bergman did a lot and a very important legacy. He's somewhat more forgotten than he should be. Things that I didn't mention is his work on gases, metals, especially platinum. I didn't go very much into mineralogy or, in fact, chemical nomenclature. I mentioned he was influenced by Linnaeus, and he actually himself developed some chemical nomenclature. Because of time, we just didn't have time to cover all these. But I hope I was able to whet your appetite, so to speak, to maybe study more about him. He deserves that. So thank you very much, and I'll be happy to take any questions.
Thank you very much, Nick. Does anybody have a question? If they do, they should just unmute their mic and ask.
Well, thank you, Nick. It was an awesome presentation. I'm really happy that I saw that, but I have plenty of questions, but I know we're limited in time, and
go ahead, let's see.
all right. So on one of the slides, I saw that somebody really criticized very much one of the dissertations of his students, or if I saw it correctly. So what was this about? Do you know what the problem was there? But did they actually try to prevent him from hiring or something like that?
Yeah, that was the previous professor, Valerius, who was the professor of chemistry, and he looked at Bergman's work on chemistry, which was not all that rich, I should say, in discovery and innovation. It was more like a review paper, and so that was one of the criticism. It's difficult to tell exactly what the criticism was. I mean, there are very few documents that are surviving, but it was partially that Bergman was not really a chemist. On one side, Valerius really wanted to have his relative or student, not quite clear, but somebody related to him. He wanted that person to be appointed, and so he would try anything to criticize Bergman to prevent him from getting. There are probably some documents that have survived. I'm not aware, but that was the background of the story, right? He was trying to prevent Bergman from becoming the next professor of chemistry because of his very small contributions at that point to chemistry.
Well, the reason why I loved that much this period of time, is because people started as universal scientists, and we went the full circle. Now we came again to interdisciplinary science, of course, on a higher level, but this is really amazing how the things developed. So yeah, thank you for bringing up to our attention this person. It's yeah, he obviously, I mean, I've never heard about him honestly before, and he doesn't deserve to be forgotten. So thanks.
Hopefully we fix that. Do you have any others or any? You said you had plenty, but
Oh no.
okay.
Well, I don't. Yes, I do, but maybe I'll get back to you on the phone.
All right.
Okay.
That works too.
Any other
Any
questions?
other questions from the audience?
Hello, Dr. Tsarevsky.
Hi.
So the slide where you showed Linnaeus's table of affinities.
Not
So,
Linnaeus, but Bergman.
Oh, Bergman. So I was wondering more of the symbology there. So what goes into the depiction of the symbols of these elements, like gold and silver, like silver being a half moon and
The half moon. I mean, these were some of these were from the alchemical period, right? So these are from the alchemy period. So the metals were assigned to various celestial bodies. Sun was gold, and moon was silver, copper was Venus, and so on and so forth. Some of these had to be, some new had to be introduced. You have so many combinations between things. So some of them were kind of inspired by alchemical symbols, but they're not alchemical symbols. And so this is the original paper or table of affinities by Geoffroy. So many of these, as you see, some are some letters when he ran out of symbols. I mean, Berzelius was later responsible for using really letters to designate chemical elements and substances and things like that. So at that time, it was very difficult to how to represent things. But all of these are actually explained on the bottom of that table by Geoffroy and later by Bergman, of course, the much more detailed. There are all definitions what these symbols mean. So some of them were known, some of them were newly introduced by Bergman, and they're all explained there.
Mm-hmm. So if I can ask one more question.
Okay, sure.
How were these analyses performed? Like, how would they know the affinities for these ions for each other, like calcium sulfate over calcium chloride, and how would they perform this measurement to determine?
This one is easy. You mix calcium chloride, which is soluble in water, with potassium sulfate, which is also soluble in water. Something crashes out of solution, precipitates. So that suggests that two of these things stick together. You analyze it, it's gypsum, so it's calcium sulfate. So apparently calcium really likes sulfate, right? That was the conclusion. So it's not some kind of thermodynamic type of affinity. You measure enthalpy, which we would do today, or some binding affinity, something of this sort, of course. It's basically observations. So, I mean, here I'll go back to Geoffroy very quickly. So this very last column, I hope you can see, the very top symbol is water. The next symbol here in the very last column is alcohol, and then you have salt. And so that's based on observation. Tells you that water has higher affinity to alcohol than to salt. Alcohol is closer to water than the salt. Salt is on the bottom. And that's an observation. You dissolve salt in water, you add alcohol to it, and salt crashes out. So because water likes to be with alcohol and then kicks out, so to speak, salt. Right? So these were the types of observations that were made. Right? So many of them are not accurate. They're not exactly affinities. Some of them are related to solubility, to other things. In some cases, you dip an iron piece in copper solution of copper nitrate or something like that. Copper is deposited on the iron. So then you conclude that iron has higher affinity to sulfate ions, as we would call them today, than copper. And that's why copper is released from that, right? So these were the types of experiments. And just very briefly, but that's what was done.
Nick, I'm going to thank you and thank everybody for listening. And we'll hopefully see you back here after the next ACS meeting in August and show you 2 more talks from the meeting. So thank both of our speakers and thank you all for listening.
Thank you.